Hi, I'm Andrea O'Hara from GeneWiz. Thank you for joining us for our talk today. Our talk is entitled Gene Editing Validation with Single Molecule Resolution Using Highly Accurate HiFi Reads. And today I have Mike Weiand joining us from PacBio. Before we get started, if we can just have a little bit of an introduction on AAV. So AAV, or adeno-associated virus, is an option for a delivery vehicle that can be used for gene therapy. Main considerations for AAV include the considerations for size. So this is limited to a range of approximately 5 kb, including our novel gene that we're introducing. And the other thing to keep in mind, main consideration, is that the overall fidelity of the AAV genome, including the ITRs, are critical for virus replication and encapsulation. So Mike, before we dive more into AAV, can you tell us a little bit more about some of the innovations that you guys have at PacBio regarding long sequencing technology?
Sure, thanks, Andrew. And so as she mentioned, my name is Michael Weiand. I'm an FAS manager. I'm covering the eastern half of America for PacBio, and I'm super excited to be here today and talk a little bit about a lot of the research that we've been doing. Over the course of our talk here today, I'll go over a lot of different things. I'll set the background to PacBio sequencing in general, and then give some granularity associated to some of the workflows and how easy it is to kind of get going and leveraging long reads for your research. And we'll dive into a little bit of the analysis and also talk about all the different research and collateral that we've seen. So as AAV gene-based therapies have entered this clinical translational phase and commercialization, we've seen a pretty high adoption in a couple of years with that. So again, I'm super excited to talk about how we can help foster those experimental strategies. And so just to set the baseline to the technology itself, PacBio is a long-read sequencing technology. We've been around for quite some time, and there are sort of 4 staples to the technology itself. We generate very long reads. Those reads are on tens of kilobases of length, and they go out super long. And we're able to sequence a whole host of different types of applications and sample types. So we sequence samples that are only a few hundred base pairs in size, all the way out to 50 or 60 kb as the insert that we're actually leveraging and trying to interrogate. In addition to sort of some of the foundational pieces of PacBio, we're able to achieve really, really high accuracy. And so I'll show a couple slides on that a little later when Andrea and I are diving into it as well. But again, we see no systematic errors associated to that, and are able to achieve QV50 values for a lot of our sequencing types. And again, specifically for AAV, one of the really nice highlights to that is we're allowed to get single molecule resolution. So we can really dive down into the positive and negative strands associated to these products and be able to identify what's in there. And then on the back end, there are some other things that suit to specific applications that can be really, really helpful based on sequence context or epigenetic information that all comes along with the sequencing itself. And so just to show a little bit more granularity to the workflow, again, this kind of layers onto a lot of different of the application spaces, and not just specifically AAV, but we can take anything that's double-stranded, and it works into our PacBio workflow. We create libraries, and then we put it on our sequencing system, so our SQL detection system. This can be done in a couple different arrays, and sample prep and library can be across the different portfolio suite that we sort of offer, and sequence a lot of these samples all at once. So I did, just in case the folks that are attending and haven't done a lot of sequencing, I did want to speak to it a little bit, right? So previously, when you've thought about it, there were 2 kinds of paradigms that you could think about. When long reads first hit the market, right, or hit sort of a decision-making timeline, you had to decide if you wanted really long reads or really accurate reads. And one would choose one or the other, and really kind of have to plan their experimental profile out associated to that. Well, over the past few years, we've really made some startling increases and done really, really nice things associated to it. So now with PacBio's HiFi reads, you can get not only reads that are super long, but that they're also extremely accurate. And so that allows you, in one data type to do a lot of different things.
Yeah, and I'm sure, as you have noticed as well as I have, there's been a lot of excitement about the HiFi technology. Can you explain this in just a little bit more detail and some progress that you've made in this space?
Sure. And so, PacBio HiFi reads are, leverage our single molecule circular consensus sequencing mode. So, people can be familiar with PacBio, but again, just to baseline some of this, we can take any sort of double-stranded material and we ligate on these hairpin adapters. The polymerase reads ourselves have become so long with the technology advancements over the past few years that it allows us to build a consensus sequence from multiple passes of the SMRT cell, leading to extremely accurate HiFi reads. So leveraging this intramolecular consensus, it allows us to go around that template many, many times and be able to do a lot of different things with your research.
And so switching gears and diving back into AAV, how can this be used as a QC for these gene therapy delivery vehicles?
So many labs over the years have been intensely focused on developing AAV strategies, right? A lot of aspects that these folks have started to be interested in have depended on long reads. So I've been around for a little while now and sort of have been helping with these AAV approaches for over 5 years. And we've seen sort of the strategies and efforts broken down to several phased approaches, whether it's discovery and just general engineering, or groups that are aimed at vector design and trying to get high quality vector QC. Others check the platforms and develop their editing efficiency and make sure that the changes that they're making are actually there, right? And making sure that they're building out some of that for safety, right? In terms of these truncation events or different things that they're going to try and package and put into people eventually. So again, one of the major advantages that we've seen is that with PacBio and these long reads, you can reconstruct these fragments in silico, right, to determine the template molecules and have everything as full length and really understand what's in there and what they've been able to package. So having that type of resolution has really helped single vector problems. And so again, we've seen this and historically with AAV approaches, there's been some trouble getting through secondary structures, right? It's been a big challenge for second generation sequencing technologies. And this can be seen whether it's in coverage dropouts or particular regions or chimeric regions. And so from a lot of the publishing and a lot of the work that we've done, we've been able to see that we don't have any trouble getting through that. And not only get through them, but again, detect and quantify ITR damage and repair in vectors and understand what's actually happening.
Exactly. So can you just talk a little bit more about some of these recent advances you've seen specifically with AAV and PacBio sequencing?
So there's been a whole host of publications, right? And I have a few screenshots here of some things that are some primary ones that I've worked on. So again, I wanted to layer that on in terms of some of the things that I have a lot of familiarity with. But smart sequencing has been the only method that can really resolve and cover the entirety of the AAV genome from the intact sequence of the ITR to ITR without the need for sequence reconstruction. And so some of these publications talk about reverse packaging of the genomes, originating it from the plasmid backbone or whether they're looking for host genomic sequence. But it's really allowed for discovery and redefining the quality control standards for this type of work. And so I'll talk a little bit about a couple different examples, right? So let's not just talk in broad strokes, but get down into some real world information. And so this is a group that over the past 4 or 5 years I've worked very closely with. They're at the Jorge Gene Therapy Center at UMass, University of Massachusetts, in Guanping Gao's group, and they were able to show that vector genomes that have been packaged that were extremely heterogeneous, and so that was something that was really interesting to start, right? So prior to that, in a lot of their work that they had been doing was with second generation sequencing technologies, and they thought that they were packaging things appropriately. And so you can see here in this screenshot, this is an IGV snapshot, an IGV display of individual reads aligned across it. This is a self-complementary AAV genome reference. And this approach really allowed them to understand the relative distribution of truncated genomes and sort of understand full length versus vector preps. And so since each one of these self-complementary AAVs are actually a linear sequence, they were able to really kind of dive in and illustrate the alignments associated to that. And so another example is from the group at the Sanger Institute. So they've done a lot of different work and a lot of different efforts over the past in trying to figure out, you know, some of their gene editing outcomes and really trying to understand what they have there. And so with this group, they leverage long reads to get across that PIG-A genome, PIG-A allele locus. So it was something that, you know, they were able to span the reads and characterize the changes that they were making and do this accurately. So if you look at the bottom left-hand side of that figure from this Nature Biotech publication, you can see the example of the alleles tracking, right? And tracking those changes and tracking those deletions that they had tried to put in part.
These are fantastic. It's so great to see examples of how scientists are leveraging PacBio in the application space. So can you tell us a little bit more about what people would need to do to get started, or just a little more about the actual workflow that you would need to use PacBio for these approaches?
Sure. And so this is something that's been great for me to see, you know, having done a lot of this work over the past few years. So early on, we spent a lot of time with these workflows and trying to build that out, and it was a little bit of a head against the wall type scenario, right? Where we really had to usher things along and really had to handhold a lot of that. And so over the past years, we've validated these workflows and started to build them out so that way it's much easier to get along the experimental path. So you'll see in this sort of screenshot here, there's, you know, procedures, there's, you know, different things associated to how you can purchase it, and again, walking through the library construction portion, it's become a lot easier. You know, in terms of importing these things into workflows that we already have established for other amplicon approaches. And so I'll talk a little bit about, in the next slide, a little bit about, you know, how to convert those different types of things. And Andrea and I will go a little back and forth, but it's been a really nice thing to see how it's evolved, and that these workflows are much more streamlined. And so, you know, in general, a lot of the questions I get are built out around extraction, right? And so whether we're, you know, doing particle or host cell purifications and how do we build these different types of things out, or what's within the experiment itself. So for the PacBio workflows, we've generally used either single-stranded AAVs or self-complementary AAVs and done taken those into our workflows. You know, there's been a lot of effort up front and a lot of publications and research, and I know that a lot of people have different flavors for this type of work, and it's been something that's been great working with GeneWiz, so I know that they've sequenced a lot over the years, and so I've been happy to see how that's evolved as well.
Yeah, and I know just based on our own customer experience at GeneWiz, we're really aware of this single-stranded to double-stranded DNA conversion step in particular. It's a difficult step, but it's really critical for getting everything prepared for the downstream library preparation. At GeneWiz, we have developed a proprietary method for this conversion. But once that's actually done, what happens next?
So once everything's double-stranded, and again, you know, having that solution that you guys have has been huge, because it's generally been a hurdle, or it's generally been something that I've dealt with where, you know, sites will either want to do it themselves or they'll want to outsource some of that information or some of that research, and it's been something that's really good to get over that hurdle. Once it's gone that far and it sort of comes into the PacBio workflows, that's where it's pretty streamlined. So we funnel it into our express template prep procedures, our library construction. It's a pretty short turnaround. It takes 4 or 5 hours to make the libraries from input material to smart bells, and, you know, having those hairpin adapters on either end. And that's pretty standard molecular biology, where we've been able to leverage all those different things that we've done on a few different places. You know, after that, the sequencing preparation and some of those other things that a service provider would have are pretty straightforward. Again, we've had a few other workflows, and so again, trying to buffer for how I would approach these types of strategy or doing long read sequencing. So this is a procedure. This is a little bit more granularity associated to it. And this would be one that we would barcode, right? So you would put barcodes in your hairpin adapters there and allow you to pool multiple samples together. And this is something that we've seen a lot of customers leverage because it makes it not only much more affordable, but then they can process a lot of different experiments to get to a lot of different discoveries pretty fast.
And so once they have these libraries all prepared, what sort of a read length can they expect when they're actually doing the sequencing?
So I've populated sequencing performance, right, of outputs. And so I talked a little earlier on that we generated super long reads with PacBio, and this is an actual histogram of what those reads would look like, in specifically for 5 kb, right? So just under 5 kb for AAV here. But you can see that over half of the reads are over 100,000 bases in length. And even though we don't need to necessarily span all that information, again, talking about HiFi and talking about the approaches that HiFi walks through, right, where we're able to go around and around, it drives us to build out a very good intramolecular consensus sequence. So you see over here on the right-hand side of the slide, I have a mock of what an actual AAV library would look like, right? So you'd have a SMRT Bell adapter on either side in the blue, and then you'd have your sequence of interest, right? Your single-stranded AAV in this case, or self-complementary, or whatever it would be in the yellow and the purple. And so leveraging that read length that's on that base density plot there, you'd go around this molecule many, many times, and that again allows you to get really good resolution associated to what you're packaging. So even if things aren't all full length, right, and you have this large distribution of what you might have created, you're going to get really good sequence accuracy associated to that. And so no matter what system you use, and you know, the outputs can be higher or lower, and you have a lot of different options for how you want to sequence and how many samples you would pull together and all those different types of things. But I have pretty good guidance on that front, which is great.
Fantastic. And so now we get to the best part, right? So we have all of our results. What does the analysis actually look like for all of this?
So my background is in wet lab and molecular biology, so I'm definitely an analysis hack here, and I think that's the nice thing associated to it. So I have a little screenshot of kind of how things work in PacBio landscape, and this is something that I'm able to do as well. You know, to speak to the ease of use and speak to, you know, some of the simplicity associated to it, right? Again, you would walk through, and a lot of this stuff can happen in less than a day, and you know, we have different guidance, whether it's on our PacBio web pages or whatnot, but you demultiplex samples that you would pull together and run them through a few different things, and then that would get you into your secondary analysis, tertiary tools, right? Things that you're familiar with, whether it's IGV or anything on that front, and a lot of that stuff is easy and pretty straightforward. And so we have repos for all this type of information, and again, the message that I want to kind of get across is that a lot of that is pretty straightforward and pretty helpful, and we've moved it along nicely.
Fantastic. Yeah, and so one question that I get a lot is actually if you're able to tell any information about what strand. So again, we were talking about these single-stranded DNAs. Is there any way that you can detect strand-specific information using PacBio approaches in the analysis or just in general?
And so I have a couple slides on that to talk about it, right? So one, to just sort of walk through, you know, some of the sequencing output or how it would actually be sequencing across, and then I'll show an example of a self-complementary AAV. So again, you know, as you're sequencing across and you're generating those, this is, you know, what our normal SMRT Bell adapter would look like, right? So you would start here with the blue, and you'd sequence around your templates and your molecule. Because it's a Phi-29 derivative, our sequencing engine, you go around and around at strand displacing, and it allows you to generate a super long read. So our polymerase read, that 100 kb read that I had showed just earlier, it would look like this, where you would have your plus strand, you'd have your SMRT Bell adapter, you'd have your minus strand, and so on and so forth. And so the analysis that we have in place for a lot of different applications allows you to do by strand analysis. So you can parse out those subreads, all of those yellow reads that have been tracked across that 100 kb read, and you can line them all up together and get an intramolecular circular consensus read associated to that. So if there are things, you know, that are non-complementary, you know, like ITRs, you know, that's or a self-complementary piece of AAV, this would be one of those things that you could do and you could run across. And so looking at, you know, a little bit more granular of what an actual output would look like, again, you have your ITRs in here, and this polymerase read would track across that whole thing, right, where you're going plus and minus, you're sequencing through all that different type, and that would be the output that you would get that you could then interrogate. And so, again, I wanted to speak a little bit more, and we'll talk a little bit on the front of all the different resources that are available. But as I mentioned, this is a repetitious slide from earlier, and I'm always a creature of habit for that type of thing. And I wanted to just show a lot of the different pieces and collateral that are there and available, right? So, most of this is all written out. It's in procedures, and it's been something that we've seen a whole host of adoption. I showed a screenshot of some publications earlier. Andrew made me parse that down. I had a giant list of things, and it was pretty ugly and over-consuming, and it was one of those things that there's been a lot of research that's happened across the landscape.
Absolutely. And so if you can just go to the next slide, Mike. Thanks. At GeneWiz, so obviously PacBio has a great system in place. At GeneWiz, we offer a full end-to-end PacBio sequencing solutions for AAV. This starts from viral particle extraction, includes the single-stranded to double-stranded DNA conversion, library preparation sequencing, and that downstream analysis. As is true with all of our GeneWiz projects, including AAV, this includes high-quality data and results with real-time updates throughout the project from our first-in-class PhD level support scientists. Of course, if you are thinking of doing one of these projects and you have any questions, we always appreciate talking to you all, whether it's PacBio directly or for these AAV projects. So Mike, before we wrap it up, do you have any additional resources you might recommend to our audience?
In addition to myself, yes. So one little pitch here would be go to the PacBio booth today as part of this event. It'll be great. We'll all be there. I'll be there both days. I have other colleagues that will be there to help. And as I mentioned, there's a lot of different resources and collateral, and I can answer direct questions associated to that. So not only do we have webinar recordings or application brochures, high-level things, and things that really get down into the weeds, there's a lot of different information and collateral associated to it, from the front end all the way through the back end. So not only myself on the wet lab side, but we have bioinformatics FAS that are well versed in this type of application and are able to help continue projects along, right? So not only sequencing, it doesn't sort of end there, and it's one of those things that we want to make sure that we shepherd everything across and really get a lot of different resources out there. So in addition to, as I mentioned, GeneWiz and Andrea talked a lot about their AAV and sort of conversion. And so this is just a little bit more granular and a screenshot to something similar that we use too, right? That you'll find on our website, on PacBio's webpage, and again, trying to layer in the ease of use for the application and trying to get you as much information as possible. And so with that, I will take some questions. Thank you.
Thank you. So Mike, I do have a few questions from our audience. The first question is, can you comment about some of the differences between short and long read technologies and what some of the pros and cons might be for each?
Sure. So we've seen a lot of people leverage long reads, right? And the primary differences that they've been able to do against some of their short read approaches have been detecting the truncation and fusions, right? And this single molecule resolution allows you to investigate these types of things and span that whole gap. Again, we're using super long reads to cover lots of things, and the fact that you can go around and around the molecule and get that consensus accuracy associated, that can give you pretty good confidence around what's in the population in the mixture, right? We've done early publications, we spiked it in on purpose, right? We had all these truncated genomes, and we were trying to have a lot of different types of things packaged to understand the resolution that we could detect. And I think that's been the primary piece to it, yeah.
Excellent. And so when you're doing these PacBio sequencing, the next question we have is, what is the recommended input material amount for PacBio sequencing?
And so this can vary pending, how big the experiment is or how small it is. The procedure and checklist that I showed earlier for barcoding, we'll talk a little bit about that in terms of how many samples you're going to pull together or how many that you're going to, run a single experiment. If you're running a single experiment, the input will be a little bit on the higher side. It'll be a microgram at least of material, so that can be a little bit heavy for a viral prep. But as you start to pull more things together or replicates or something, those inputs reduce, right? So if you were doing 4 samples, it would be a couple hundred nanograms per, right? And you match up to that 1 microgram lower level.
Excellent. And then one last question I have here is regarding CLIA or GLP. So if you're sequencing for CLIA or GLP, is that the same or any differences when you're doing PacBio sequencing at that level?
Well, you'd also know a little bit more on that front than me. No, but the libraries are transferable across all that type of stuff. So once you've made that library, you have the option to do research-based or do a few different things. And as I mentioned earlier, there were a couple different systems that you have offerings for, right? So high throughput, low throughput, and all that type of stuff. So you shouldn't run into anything once you've made libraries associated to it. But some of the other requirements, and again, why I send a lot of folks to GeneWiz is because you guys know how to do it really well. Yeah.
Great, thank you. All right, those are all our questions for today. It's been great having you with us. I'm so glad we had the chance to chat. And thank you everybody for listening in as well.
Yeah, thanks everybody. This was great. It was an awesome opportunity to have, and we really appreciate it. And so my last little pitch for the booth too, right? Come ask questions. I'm happy to help if I can.